Welcome, folks, this morning. Our talk this morning is from Dan Runcie, professor at Department of Plant Sciences at UC Davis. He'll be talking about genomic predictions from hundreds of trials of it. Looks like maize. Dan?
Hi.
Take it away.
Sure. Okay. Well, thanks for coming. So the talk that I'm giving is on our work with genomic predictions. We've been working with the genomes to fields data, and largely a lot of this work is done by my postdoc Hai Xiao, and so acknowledging him at the beginning. So we want to talk about how to do genomic predictions when you have hundreds of trials, and you want to sort of leverage the variation in genetic relationships across all these trials in a way that uses all of the information available to make all genetic predictions better. So one of the fundamental challenges that breeders face is that there's no best variety that's best everywhere in every field and every year. That's because different fields have different environments, and genes that work well in one environment may not work well in another. So you may have seen this picture before. This is from a different project we've worked on in the lab, looking at local adaptation in maize to extreme highland environments in say, Central America. And these pictures show two corn varieties grown next to each other, one in a lowland field in Mexico. And so in this case, you can see that the highland accession is not growing as well and makes puny ears. But if we take these same two genotypes and we grow them in a highland field, the opposite is true. So with the highland ones growing faster and the lowland ones making puny ears. So this is an example of what we call a gene-environment interaction, or G by E, where different varieties perform best in different environments. And because G by E's are so common, breeders need to develop varieties that are targeted to specific regions, and we call these the target population of environments. And so in order to do this, and be able to breed for different locations, breeding programs need to invest in multi-environment trials to compare varieties across a range of conditions. This figure that I'm showing here is from a paper that I took by Gaffney et al. About a Corteva trial. And so each dot on this map is an on-farm trial comparing hybrid corn. And so they ran over 10,000 trials in this study. However, these trials are complicated, expensive, and difficult to analyze. And so do we really need to run so many trials? And if you do, how do you analyze these data? And what can you learn? So these are the types of questions that my group has been working on from the statistical standpoint, and I'm going to be talking about some of our methods and how we're doing that today. So I don't have access to a data set like Corteva's there. So instead, we've been using this large public data set from the Genomes to Fields initiative, which I'm sure you've had other talks about. This is not as big as the Corteva trial, but it's still quite large. And so in this trial, we're working on data from over 3,000 hybrids and almost 200 trials. And so that's about 80,000 plots of data. These trials come from 20 states, 42 locations over 7 years. And I'm going to be focusing on grain yield and flowering time as phenotypes collected in these trials. We also have whole genome genotype data that we can use to build the genomic predictions. So our goal is to ask this question: can we build a genomic prediction model that can predict the best hybrid anywhere in this, at least this part of the US? And so that's the overall goal. Now, a major challenge here is that in some ways, this very huge data set is still very incomplete. That's because not every hybrid can be grown in every trial. And so that's what I'm showing in this figure on the left. So here, this is basically a big figure of a matrix. Each row represents one of the 3,000 hybrids. Each column is one of the 200 trials. And I'm filling in the cell there if that hybrid was grown in that trial. And so you can see that it's still, that it's very incomplete. Most hybrids were grown in only about 20% of the trials, and most trials have only about 10% of all hybrids. So if we want to compare hybrids' performance in every trial, we're going to have to impute all of these missing cells. And we want to do this in a way that is sensitive to all of the gene-environment interactions that we expect to be going on between the different trials. So those imputations are accurate and make for valid comparisons. So this is a type of problem that my lab has been focusing on for several years. And the R package that we developed called MegaLMM, we think can do this type of a imputation quite well. So MegaLMM is designed for doing genomic analyses across thousands of traits at a time. And we can think of the performance of a variety in a single trial as a trait. So the main goals of this method are to be able to simultaneously use data from all hybrids in all environments, to flexibly learn which trials are similar and which are different, and to do this in a way that can be capable of fitting data from thousands of lines in tens of thousands of environments potential, like that Corteva trial, in a computationally efficient way. And this has been a challenge before, and this is really several orders of magnitude bigger scale problem than other methods have been able to deal with in the past. But it seems to work. So this figure, where I'm comparing the accuracy in which we can effectively impute or predict based on the genetic data and the relationships among trials, the grain yield in each of these trials. And so in this figure, I'm showing the average prediction accuracy averaged over about almost the 188 trials. In blue is our method, MegaLMM, and then in red and green are two sort of reference methods that we're using. So the red line, the red bar, 100%, I'm calling it 100% G by E. What I mean by this is we model each trial separately. So we're not learning, we're basically assuming that the performance in each trial is completely independent, and so there's no sharing of data across trials. Going in the completely opposite direction, I say call it a method no G by E, by which I mean we effectively average all trials together, make a single prediction of the relative performance of each hybrid, and then use that same prediction in every one of the trials. MegaLMM is basically an interpolation between these two extremes. So we model the trials, as I said, as correlated traits, and then what we do is called partial pooling of information across trials. And so we learn which trials are similar and which ones are not, and use that information to share information and make better predictions each trial. And what this graph shows is that it's generally fairly effective at doing that. So the other two approaches averaged about 25% accuracy, while MegaLMM is about 38, I think. I don't remember exactly the number. Now, I'm just showing the average here, but you should always be suspicious of averages because they can hide a lot of variation and give potentially misleading image if, say, MegaLMM just does spectacularly well in 2 trials, but poorly in all the rest. That may drive up the average. But that's not what's going on here. So here, each point is now a single trial, and I'm comparing the MegaLMM accuracy on the y-axis to one of those reference ones, the 100% G by E on the right. And so a point above the line is one where MegaLMM does better, and you see that it does better in almost all of the trials, often up to 20 to 30% better. And so if a 20 to 30% increase in accuracy, we can do sort of by partitioning data in trials, and that's like doubling the size of the trial in terms of the number of plots that you've grown. And so it's a pretty significant improvement by using a model that's flexible enough to incorporate all of the data at once and try to use that effectively. So that shows that MegaLMM is effective. And so the rest of the goals for the talk, I want to show you sort of how MegaLMM is able to do this, and what can we learn from doing that. So I'll start by discussing some strategies that we use for modeling G by E. Take that into how can we take some strategies from mining big data and genetics to do this better. But then I want to take a closer look at the results and ask whether they're really as good as they seem. And finally end with some thoughts on where we should go from here in trying to improve breeding for gene-environment interactions. So those are my goals for the talk. Now, broadly, there are 2 approaches for modeling gene-environment interactions in quantitative genetics. There's what we call the reaction neuron model and the correlated traits model. These 2 approaches look at gene-environment interactions in very different ways. And I think the contrast between them is informative about modeling G by E in general. So let's start with the simplest approach, where we have two fields, call them north and south. Now for the reaction norm model, we plot the performance of each hybrid in each field. So I have two hybrids here, pink and green, and so we can plot their performance in each field and then connect them with lines like this. Since the lines aren't flat, we see that each hybrid's performance changes between the fields. That's what lines are not flat means. We call the slope of this line the plasticity of the hybrid, and the difference in slope is the gene-environment interactions, or how different their responses are to the change in environments. Contrast that with the correlated traits model. For the second, here we consider this experiment to be measuring two separate traits. Yield in the north field is one trait, and yield in the south field is another trait. Because they're different traits, each one gets its own axis. We don't put them on the same axis. And so if we have two different axes, we can make a scatter plot of the two yields of each hybrid. So on this graph, if we add the one-to-one line through the middle of the graph, we can see how much each hybrid's yield changed between the environments. And so that's how we see G by E, like that. So the reaction norm model is effectively a model of the plasticity of each line, while the correlated traits model is a model of the similarity of the two environments. Now, the implications of these two approaches start to show up when we add a third environment. So how do we fit this third middle field into each environment? Now, in the reaction norm model, we could place the middle field here in the middle of this graph and then predict its yield by projecting along these reaction norms. That's pretty efficient because we only have to measure two parameters per line, the slope and the intercept. And then we can project yields at any number of intermediate locations. In contrast, adding the third field in the correlated traits model is a bit more difficult because now we're adding a third independent axis, and we need to add two additional scatter plots here. But it's not clear how to fill these in. How can we predict if these correlations are going to be strong or weak or even positive or negative? Now, the easiest way to do that is if we have data on other lines in each of these three environments. If we see that yields are generally negatively correlated between the south and north field, but positively correlated between the south and the middle field, we can then make projections of our pink and green lines based on these correlations. But to do this, we need data on a lot of lines in each of these environments to do this. Clearly, this is more difficult. So it seems like the reaction norm model is a clear winner. But there are problems with the reaction norm model. First, was it really reasonable to place the middle field in the middle of this plot? Yes, it's in the middle geographically, but plasticity is controlled by the environment, not geography. And maybe right for temperature to put the middle field on this axis, but maybe it's wrong for soil depth or some other environmental variables that might be relevant. And so how many environmental variables are relevant here? And are the reaction norms, these lines here, really linear? The reaction norm model is pretty limited once we add more complexity like that. So the correlated traits model can potentially overcome some of these issues. So unlike the reaction norm model, the correlated traits model is a very empirical model, meaning that it's much more data-driven. We don't really make any assumptions about some trials being intermediate between others, or any assumptions about which environmental variables are most important drivers, or whether the plasticity along any environmental variable is linear or not. Instead, we just let the data dictate the patterns. Sorry, my slide's behind. The problem with it, though, is that the models start to get very big with very many trials. As you add trials, we then have to add scatter plots here. And that means we have to estimate the correlations between every pair of trials. And when you have a lot of trials to estimate, that's a lot of correlations to model. So anyway, so which approach should you use? The reaction norm model is very appealing because its calculations are easy, efficient, and there's lots of statistical software for doing it well. However, it only works if you know what environmental variables are important. The correlated traits model doesn't require any extra information, but can be extremely computationally efficient. Computationally intensive. So given this setup, what does MegaLMM use? Well, MegaLMM is essentially a correlated traits model, but it approaches this by kind of fitting a reaction norm model under the hood by making guesses at what environmental factors. So it's kind of a hybrid. So now, to go a bit deeper, I want to take a little sidetrack into machine learning or data science. So essentially, MegaLMM needs to estimate each of these correlations, actually twice, once for genetic information and once for environmental similarity. And each of these correlations is a parameter. So with 5 trials here, we need to estimate 15 parameters. If we had 10 trials, that'd be 55 parameters, but we have 188 trials, which means we have almost 20,000 parameters to estimate. And we don't have enough data to do this directly or based on the data themselves. Now, sort of a general law in statistics is that the more parameters you have, the less precisely you can estimate each of them. And the more parameters you have, the longer your computation takes. And when you have more parameters and observations, models generally can't converge at all or give completely nonsense results. However, there are lots of approaches in modern statistics and machine learning for dealing with this problem, which we call the P is greater than N problem, meaning more parameters than data. For example, genomic prediction models may use data on 50,000 or maybe 10 million or 100 million SNPs to predict phenotypes. Each of those SNPs in these models needs its own parameter, yet they work with data from only 200 lines or so. How do they do this? Well, they make a couple assumptions in the models. They code those assumptions as in various statistical structures. So one assumption they make is that most SNPs have very little effect on a trait, which means that most of these effects are very small. There may be a few large effect SNPs, but they are very rare. And so they code these, and what this is called is doing regularization or Bayesian shrinkage. And this is the approach that tools like gblup or RRBlup or Bayes-B or any of a number of the machine learning approaches are effectively coding assumptions like this into their models. So what we'd like to do is take this same idea of regularization to our estimates of the correlations between traits. So to state this problem again, from these data, we want to take our data, our observations of relationships among the trials, and turn those into these estimates of the correlations, which we can represent in a matrix like this. So our goal is to get regularized or more efficient estimates of these correlation parameters. Now, the assumptions that were used for genomic prediction are that most SNPs have very small effects, which means so that the coefficients, the parameters of those models are mostly small, and large effect ones are possible, but are rare. Now, if we just take these same assumptions and translate them into what they would imply for the correlation parameters, we would get that most correlations are close to 0 and large ones are rare. But neither of these assumptions are at all reasonable for modeling correlations among trials. So most correlations are not close to 0, and large correlations are not rare. That's because many trials are highly correlated. When you have trials in similar environments, or say the same trial in multiple years, we expect them to be highly correlated, especially when they're in similar environments. So these particular assumptions don't work, but we need to come up with other type of assumptions where we can sort of get the same effect of regularization, but where we regularize towards large correlations instead of small ones. So how can we come up with these assumptions that are biologically reasonable? The way that we do this in MegaLMM is by effectively making up environmental correlates. Remember, we don't actually know what environmental factors are relevant for driving gene-environment interactions or how they differ among trials. But say that we did. Say that we knew that temperature was the key factor. And so we can measure the temperature in each trial and arrange them say from the coldest to the warmest like this. Then we could predict, the correlation or covariance matrix, P, across the trials by simply taking this vector of temperatures, multiplying it by itself, and it would turn into this matrix like this. And so what this matrix means, say blue is colder or is low values and red is high values. Red is high values. I got these wrong. But anyway, what we can see in the covariance matrix on the right is that the two colder trials have similar values. The two warmer trials have similar values, but the top to the bottom trials are very different. And so they're negatively correlated. So that's what we could predict that the covariance matrix would look like this if we knew that temperature was the driving environmental factor. But we expect there's going to be multiple drivers here. So say soil depth was also a driving factor. We could similarly measure the soil in each of our trials, rank the trials based on them, and then create a covariance matrix for soils. Now, if these two were the drivers, then what you would do is you would simply add up those two sources of covariation into a total covariance matrix. And so the idea here is that we get this covariance matrix from actually just the vectors themselves. And so that's a reduction in the number of effective parameters, which gives us more efficiency. So to make this more practical, what we can do is stack our vectors like this. And this is what's called a factor analytical model. And it's been used for modeling gene-environment interactions for a long time. So generally what we do is we combine these vectors into matrices to simplify the calculations. And we can allow any number of these environmental covariates, which simply get added together to get our final model of the covariance. The key idea here is we don't have to know what the environmental variables are important. We just observe how trials are related, and the model figures out these candidate covariates for us. Now, the new part of MegaLMM is that we encode on top of this factor structure a few new assumptions. We assume that most values in these columns, these vectors, are small, large ones are rare. And then also that we can order these columns from most to least important. And this latter assumption is what really allows us to scale up the factor analytical model to very, very many trials and also quite a number of environmental covariates. And so that's what gives us the regularization that we need to be able to apply our model efficiently to large data sets. So that's the intuition behind how MegaLMM works. So now I'm coming back to our case study of the genes to fields data set. So as I showed in the beginning, MegaLMM greatly improves the prediction accuracy of grain yield in almost every environment by learning these environmental covariates and then using that to partially pool or share information across the trials. And it's not just for grain yield. If we apply that to flowering time, we see a similar benefit of MegaLMM. We get in both of these about a 15% improvement in accuracy and higher accuracy in almost all the trials. And so the lesson from this is we should be building models that can use all of the data accessible to us at once in a way that's flexible enough to learn individual patterns, but share information when possible. So I think MegaLMM shows a lot of problems. But is MegaLMM really modeling G by E successfully? So I want to look more closely at these results and ask this question. So I'm going to look at two criteria, which I think are important. So we see that MegaLMM has much higher accuracy than these two reference models. But is this benefit really from its ability to make predictions accurately in each of the specific trials? Or is it just better at making a global prediction? Now, it could be that the individual what MegaLMM is really able to do is sort of pool information more effectively across the trials, taking advantage of the sort of the sparseness of the matrix in a better, more coherent way than the green bar here. But it's really just making the same prediction in every trial. So that's one question. The second question is, can we actually predict new trials based on their environment? And I think that the second criteria is really important if we want to claim that this is modeling gene environment interactions. We really need to be a function of the environment. Anyway, so to ask the first question, what we can do is we can take the MegaLMM predictions and replace them with the average of the MegaLMM predictions. So MegaLMM is making a prediction in each trial, but rather than using that one, we can just average the predictions across all of the trials, and then see if that average prediction is as accurate. And it's not. So the purple bar here is taking the prediction of MegaLMM in each trial, averaging them, and then using that average in each trial. Just in some sense, it's very parallel to the green bar. And so we see that it does a little bit better than the green bar. So our tool is able to get a better average prediction for yield, but that's not nearly as good as its actual specific prediction in each trial. So we are successfully able to make predictions unique to a particular trial. So that's good. That means that we are, in a sense, modeling gene environment interactions. However, do these results really show we're accurately predicting G by E? And I'd argue probably not. What I'm showing is the accuracy of predicting some hybrids in a trial based on measures of other hybrids in that same trial. The way that we assess this, the way we measure our accuracy, is that we take our data set, which you remember is very sort of patchy like this, because only some hybrids are grown in each trial. We mask some of those, we hide them from the model, and then we fit the model to the light blues and use them to predict the values that have been masked, and then measure how accurately we predict them. So we use a 20% holdout for cross-validation. This is commonly called the CV2 method in breeding, and it measures how well we can predict in each of these 188 specific trials. And so we can see is, MegaLMM is able to do this pretty well. But if you think back to this map, these 188 trials cover a very small percentage of the corn growing areas in the country. And we haven't seen any of these hybrids in the vast majority of fields. So if we wanted to know about fields in new locations like these pink dots here, or even in the same location but in the future years, this number that we get from CV2 is really not relevant. So yeah, these numbers that I'm showing here are not the right accuracies. They're not telling us whether we're going to be accurate or not in the pink dots. They only tell us about those tiny little black dots on the map. So how can we actually make predictions into these new environments? Well, fundamentally, the way that MegaLMM is built, because it's built on a correlated traits model, we can't use MegaLMM to make predictions into locations where we don't have any data yet, because we don't have any basis to learn these correlations. Instead, we need to somehow build into MegaLMM a way to use physical characteristics or weather characteristics, something like temperature or precipitation of these new environments, or maybe even geographic distance, to be able to predict what those correlations are. The problem, as I said, is the MegaLMM is a correlated traits model, so there's no way to input these physical characteristics of the new environments. For that, we need a reaction norm model. So our recent work, and the work that Haixiao has been working on, is to turn MegaLMM back into a real reaction norm model. And so the way that we're doing that, as I've described, MegaLMM basically learns the correlations among trials by effectively making up these environmental covariates, which we call factors. So what we can do is we can collect a large set of environmental variables and form these into candidate factors. For example, in the genomes to fields data, the groups that provided the data collected data from 10 different environmental variables at 20 points during the growing season. They collected soil variables, 30 of them across the site years. And then we also have genotype data on the testers that were used to create the hybrids. And so we can form these into hundreds of candidate environmental variables. I'm just giving two examples here. Say we have actual temperatures and the actual, some sort of a soil measure for each site. So what we can do is we can take our estimated factors on the left and do a regression, regress those onto our candidates. We do this internally in the model. We put these candidate environmental variables as priors for the factors, and then use those to learn these weights. It may learn, for example, that the first factor, lambda 1 there, is primarily driven by temperature, and the second factor is primarily driven by soil depth. But once we've learned these weights on these candidate environmental variables, we can then use them to predict into new environments. And so that effectively turns MegaLMM back into this reaction neural model. And so we're hoping to get the best of both strategies. Now, to see if it's working, we need to do a similar masking strategy. But this time we're going to mask whole sets of trials, not just parts of trials, not just some hybrids in each trial. So in the first test that we did, we broke the sets of trials down by year, hid all the trials from a single year, and then used MegaLMM to predict into those completely masked trials. And so here are the results. The blue is MegaLMM's prediction accuracy into these new trials, and the green is what we, our reference again. I'm going to use the no G by E reference because we don't have any data of the in this mass trials to make a specific prediction to that trial. So what we can do is we can use years 2018, 19, and 20, find the average, global average across all of those, and then use that single one in each of these, each of the individual trials in 2021. That's the green bar. MegaLMM, though, because it can use the weather and the soil and the tester data in each of these new trials in the 2021 year, it can do better. And we show a quite significant improvement in accuracy into these new trials. And again, these accuracies are broadly. They're in every one of the trials, almost every one of the trials that we hold out, we can do better. So that's good. But if we compare these accuracies, treating them as unseen trials, they are lower than for those same trials if we had seen data in them. And so the red bar is that CV2 number that we had shown before. The CV2 number is what people normally report for genomic prediction accuracy in G by E. And what we're showing here is that number is too high. The actual accuracy is lower than that, as you'd expect, because we don't have any actual performance data in these trials. So the results are, I think, still promising. We are getting a benefit. We get more accuracy into the new year, but not as much as you might have thought given the CV2 method. But this is not completely satisfying yet, because predicting into a new year, the environments, the trials in each of the years are basically in the same location. Maybe not exactly the same field, but they're very, very close together. So, and they also tend to use the same testers, the same hybrids each in consecutive years. And so these numbers may still be inflated over what you might get if you were really testing new hybrids in a completely in a new farmer's field. So so we did another type of a set test. In this case, what we did is we masked out all of the trials in a single state. In this way, when we use those, say, for example, Iowa trials to evaluate our predictions, we're not able to draw on any information from even nearby trials in our data. And so really our predictions have to be purely based on the weather and soil information. And so when we do this, the accuracies are okay. They're greater than 0, but the benefit of MegaLMM comparing blue to green is much less than it was when we were predicting a new year in the same location. So yeah, so while it looks like we're being successful, we are we're making somewhat better predictions using that weather and soil data, but not a lot better predictions. However, a surprising thing that we noticed when we looked at these results closer is that most of the benefit that we're getting from MegaLMM in these two contexts, so again, comparing blue to green, is not from making specific tailored predictions in each environment. Instead, if we take those MegaLMM predictions and average them into a single future prediction, say a single prediction for 2022, or a single prediction for all Iowa trials, we do just as well, or almost as well, as if we had made a tailored prediction for each of those trials. And so that's the purple bar. So the purple bar is again, parallel to the green one. Again, both of those, we're making a single prediction for all of the trials in the new year. We're not tailoring it for the environment of those specific trials. And we do just about as well. If we're going to new year, almost as well. If we're predicting to a new state, we actually do even better. So this is kind of confusing. We see MegaLMM benefits, at least in the CV2 context, by learning trial-specific performance. But we get little benefit of these trial-specific predictions if the trials are new into a new environment. And so, while MegaLMM is getting us accurate predictions, I think these results are saying that we're still not really effectively modeling the environmental drivers of G by E. So it's useful, but still not doing exactly what we want. So why not? Why is modeling G by E still so hard? I think there are two key factors. There's data limitation, and then there's biological limitations, partly in this data set. So by data limitations, I've slightly distinguished from modeling limitations. I've shown that MegaLMM is capable of making G by E predictions with some accuracy. And when we use some simulations that I haven't shown, we can show that if the environmental drivers are strong and in our data, that it can work. However, if we think about it, even though the genomes to fields data is an enormous project, it's probably still way too small to be able to do this effectively. So remember, we're using data on hundreds or thousands of different hybrids to make genetic predictions in each of the trials. But we only have 200 trials representing only 40 locations to try to learn the relationships with environmental variables. And that's probably not nearly enough. We have hundreds of candidate environmental variables, and to learn the relationship, to learn these weights from only 40 trials is just asking way too much from our models. We're probably not going to be able to get good models of the effects of environment until we have at least thousands, maybe tens of thousands of trials, really on the same scale, at least, of our data on the genetic side. And then there's biological limitations. So I think there's also just not enough gene-environment interaction in these two traits to really matter for these trials, for several reasons. One is that all these trials are really fairly well managed. There's a couple sort of stress type trials in there, but most of them are not. And we might expect to see more gene-environment interactions in those really stressful, or in the contrast with those really stressful trials, and just not in these data. Also, these trials include a very wide range of genotypes. They're not all elite varieties. And they include some always poor performing hybrids. So if you have a data set where you have some hybrids that are always good and some that are always bad, that means there's just, when you're looking across the whole data set, going to be strong positive correlations across all your trials, and there's not going to be a lot of G by E to worry about. And so a model that spends a lot of time trying to model G by E when it's really a small component to the overall variation is not going to look very good. So I think that if we apply this to, say, more of the variety testing stage, we might start to have better success. At the same time, though, we know that our model is incomplete. There are many factors that are unique to each trial that aren't captured by soil and weather variables, things like disease or management or other things about that. And we just don't have data on these, and we can't predict them if we don't know about them. So we're going to have to wait. So our predictions just work a lot better if we've actually seen performance in these trials, like CV2. And in that context, the model works a lot better. So these are areas that I think are worth exploring more. So to summarize, MegaLM works. We can model hundreds or thousands of trials at once, and we always get better predictions when we add more trials. And we get those better predictions because we're able to learn correlations among trials, especially when the trials are incomplete and only contain a portion of the hybrids. And so in these cases of sparse testing, we're able to basically impute the full yield or performance matrix, and that allows much more interesting modeling downstream. But I do think that most of the studies that look at G/E prediction report statistics that are really inflated relative to the actual use cases that they're trying to model. And so I think that careful study design is needed to test more robustly whether these models are actually doing what we claim that they're doing. So this work is led by Haixiao, my postdoc, and in bold are several other collaborators that have worked on aspects of MegaLM, and then other people in my lab that have also working on MegaLM related projects. And so the data that I've shown is all from the Genomes to Fields initiative, and the work is supported by USDA, NSF, my department, and Artificial Intelligence for Food Systems, which is a USDA NSF institute led by Davis, but with several other universities that I'm also a part of. So thanks for listening, and I'm happy to discuss if you'd like.
Thanks, Dan. Folks can either raise their hand or put questions in the chat. Dan, I started off with one in the chat. You made the claim that here in the G2F, the G/E doesn't matter too much, partly because of extreme lines. So I guess the first question is, how does that change if you drop some of the extreme lines? And are you, I guess I didn't word it very well here, and are you able to actually estimate those variance components? And when you say G/E doesn't matter, I guess, what do you mean by doesn't matter? What percent of the genetic variance is due to G/E?
Yeah, so we don't in MegaLM, we don't construct the model in a way that the overall some overall variance component is a parameter. The overall amount of VG and G/E is not a parameter of MegaLM. We can try to construct that parameter sort of ad hoc afterwards, and I haven't done it with this data set. I guess it'd be we could try to do that. So G/E is not a parameter of MegaLM. In a sense, MegaLM's parameters are correlation, and G/E is lack of correlation. And so we could construct some number as basically 1 minus the correlation. But what I see is if I plot the correlation across all trials in either yield or flowering time, the correlations are all estimated to be very high, in the 0.6 and higher, I believe, with the exception of a couple outlier trials that are lower. And so that's at least some ballpark guess that something like 60-70% is the typical correlation estimated from the model in terms of the genetic correlation. The phenotypic correlation is a little bit lower than that, but largely because of measurement error.
And if you drop the extreme lines, does that change?
I haven't done that. It's hard because of the sort of the sparseness of it to be able to drop a lot and still have enough to be able to test with. Something we could try to do.
Yeah. There's a question from Jin Lang in the chat. Can you model related traits, not necessarily all yield traits, for example, yield components or time series developmental data?
Yes. So in principle, yes, you can throw in anything that any traits that you want. In practice, constructing it in a way, so there's two aspects to that. So if your goal is actually yield, and you throw in other traits, there's two ways I can see throwing them in. One is for a line that you have a measure of yield in your data set, you can also append to it measures of flowering time and lodging, and I don't know what else you would put in. You put in some other variables for those same ones. That doesn't tend to give you a lot of benefit. Yield is, in this data set at least, heritable enough that adding in additional traits that you don't really care about just to give you better predictions of yield doesn't work very well. The other way I could see about doing it is you might have some trials or parts of trials where you don't measure yield, but you do measure some other things. You fly a drone and get some sort of a spectral reflectance on that. And so you might be able to do that for many more lines, and you're able to measure yield or maybe some downstream quality trait. And in that case, adding those related traits can help tremendously. And so we know it can help tremendously, but measuring how much it actually helps is actually really tricky. You can if you just look at the correlation between predictions and observations, you greatly inflate. You get a very overly optimistic measure of how well you're doing. And so we've worked some with ways of correcting that, but it's hard. So I mean, the simple answer is yes, you can throw everything in there, but the more complex answer is that there's different ways of thinking about adding them in there, and while the ones that work well, it's hard to tell if they actually work.
Maybe somewhat related, Amber's question in the chat is, it looked like model performance was better predicting flowering time than yield. And why would that be?
I think it's generally more heritable and less G by E. So more heritable means that it's just easier to predict largely from genetic data. Less G by E means that for any particular trial, there are more trials that are highly correlated with that one that the model can sort of find and pull that information over. So one way of thinking about what it's doing is it sort of takes all the trials and partitions them. It doesn't exactly do this, but the way to think about it is it partitions them into clusters of ones that it thinks are similar. And then it takes an average of those local clusters. So with yield, it may have to make more of those clusters. And so each individual cluster is smaller and has less data. While for flowering, because it seems to be more stable, overall the correlations are higher, it can make fewer clusters and then so average more trials together and thus have more data per average. At least that's my assumption.
Ruben, you had a question? Go for it.
Yeah, Dan. So if they asked you to redesign G2F, what would you do? Maintaining same cost, right? Would you drop locations? If you drop locations, which locations? Would you increase the number of hybrids that are repeated across locations? What would you do?
Well, I would use the same tester or reduce the number of testers. We find that in this data set, the genetic correlation between different testers is very low, much lower than we've seen in other data sets. And so that means it's just very hard to use different testers, compare data from different testers. And so we basically have to partition our data by tester, and it makes the whole thing a much smaller data set. And so that'd be one thing. But more generally, I think that, yes, a little bit less sparsity in it. So more years with the same hybrids would probably help. Right now, each hybrid is basically run for 2 years. Doing a little bit more there seems like it would probably have given us more signal, maybe slightly on the too sparse side. And then, yeah, I don't know. I mean, I think as I've thought more about it, I'm sort of unclear on what the application of a trial system like this might be. So there's early stage testing where you want to test a lot of varieties, and there you're sort of interested in the general performance of these, and having tons and tons of trials of sort of evaluating specific performance in each of these early stage things is maybe not so important. You're just trying to get some better candidates to then take into later stage trials. It's a later stage trials where you really want to learn the environmental dependencies of them. And there you're going to have a smaller number of varieties grown in a much wider range of environments. And it seems like the genomics field is somewhere in between. It's testing a lot of things in a lot of environments, but not enough things in enough environments to be able to run the environmental dependencies, but not enough things in any particular environment to really learn the genetic relationships in those environments enough. And so it's sort of this weird hybrid of those two stages of a breeding program. And so I think a company wouldn't do a large commercial program wouldn't do something like this. It would choose one of those two applications, which is probably the right strategy for a real breeding program itself.
Cool. Thanks.
Maybe one last question, Dan, along the lines of your concern that we don't really know the environment. The way you're modeling things, if say temperature really is the only thing that matters, does it actually make things worse by including a whole bunch of other environmental factors? Or is it MegaLM able to learn that those others don't matter, assuming you have temperature in the model?
Yeah, clearly it makes it a little bit worse. But I mean, we're using more or less the same statistical framework as GBLUP or RBLUP. And, yes, adding useless markers, if they're really useless, if they're really just noise, it will pull everything down a little bit and reduce your accuracy, but not that much. And that's my guess here, is that it doesn't probably do too much more. And we don't tend to get really worse predictions by putting in more predictors. And that could be that none of our predictors are very good, but
yeah.
Yeah.
Okay. Well, thank you very much, Dan. We're close to coming to the hour. We have another talk next week, folks, from Shanghai at Cornell. So we will see you next week. Thanks all. Thanks, Dan.
Yep.